Hi there, it's Mrs. Vance again, and we're starting on notes today on Unit 10 about genetics. Most of the information in this unit is contained in Chapter 9 in your book, which is titled Patterns of Inheritance. There is going to be a fair amount of vocabulary, as usual. Some of these terms you may already be familiar with, others you may not be. This is just kind of an overall list, and we'll talk about terms as we get to them in the notes. Part 1 is about Mendel's laws. You probably have learned some things about Gregor Mendel in middle school. We're just going to do a quick recap about what he did and how he taught us about how things are inherited. So he was a monk in Bavaria, and he began experimenting. He was in charge of the garden, and so he began experimenting with garden peas in the gardens there at the monastery. As we already know, heredity, of course, is the transmission of these traits from one generation to the next. And the scientific study of heredity, of course, we call genetics. And Gregor Mendel is the one that kind of started this whole thing. He didn't really call it genetics at the time, but he began in the 1860s. He was breeding garden peas. He looked at traits that were present in the peas, and he did crosses to see how they were inherited and things like that. He wrote a paper that was not widely distributed at the time. It was kind of forgotten about for a while and was rediscovered in the early 20th century. Anyway, he argued that parental organisms pass on their offspring to their offspring these heritable factors. He didn't really know what they were, but they were things that retained their traits from one generation to the next. We call it now a heritable feature that varies among individuals, like flower color, which is one of the first ones we'll talk about, is called a character. And the different variants, the different expressions of those characters is called a trait. And so we'll talk about various terms for those as we go through the unit. So he was working, first of all, with true breeding varieties. These are ones that when they're allowed to self-fertilize, self-pollinate in the case of peas, they're going to produce offspring that are identical to the parent. What he did is he cross-fertilized true breeding varieties for different traits and produced hybrids. These are the offspring of two different varieties. Okay, and so this cross-fertilization is called a hybridization or genetic cross. We're going to call the true breeding parental plants the P generation, P for parent, and the hybrid offspring are going to be the first generation, are going to be the F1 generation. And then if you cross two F1 plants with each other, you're going to get an F2 generation. And those are terms that we'll talk about over and over again as we progress through this unit. So make sure you keep track of what's P and what's F1 and what's F2. All right, so these are the 7 traits he looked at in garden peas. The shape of the seeds, whether they were round or wrinkled, the seed color, whether they were yellow or green, the shape of the pods, whether they were inflated or constricted, the color of the pods, the color of the flowers, the location of the flowers, and the plant size. And you can see here represented in this diagram the different traits and what they are, the letters that we use when we're doing genetic crosses to refer to those different traits, and which ones are dominant, which ones are recessive. The top row, of course, with capital letters are the dominant traits, and the lower row with the lowercase letters are the recessive ones. Those are the ones that are going to be hidden if there is a dominant trait present, a dominant allele present. So first of all, he did a monohybrid cross. We're looking at two individuals that differ in a single characteristic, okay? And the first one that he did, that we know about, was crossing a plant with purple flowers with a plant with white flowers. And the way he did this is he actually cut off the stamens, which are the part of the flower that produces the pollen of one flower, and transferred pollen from the other one to the pistil, which is where the fertilization occurs, of the other flower. And he did this not with just one plant, but with multiple plants, multiple flowers on multiple plants. When he did this cross, then all of the peas that were produced from that first cross, when they grew into plants, had purple flowers. When he crossed some of those with each other, Then what he found is that F2 generation had 3/4 of the offspring produced purple flowers and 1/4 produced white flowers. And that was not really what he expected to see. Okay, so we're going to look at what happens in the gene. So the trait that we're looking at here is flower color. Let's say the gene, it contains this sequence that is shown here, ATG, CGAT, TCCA, and that produces purple pigment in the flowers. The white flowers have a similar sequence with that 4 base difference there, TAAC instead of CGAT, and those produce white flowers. So we're going to call the allele, the form of the gene, capital P, and the for purple and lowercase p for white. The alleles are the different versions of the gene. And so we'll use that term a lot to refer to different traits. Another one that he looked at was pea shape. Okay, and so he crossed plants that produced round peas with those that produced wrinkled peas. Okay, and again, you can see the difference here in the DNA sequence. That's the gene. Okay, the allele would be capital R for round and lowercase r for wrinkled. And again, the alleles are the different versions of the gene. And so what he found was that when he did crosses of those, he found different proportions of the offspring that had different traits. So he came up with, as a result of all of his experimentation over a number of years, working with thousands of pea plants and thousands of peas, he developed some hypotheses, and we're going to use these today. The terminology that he used at the time was different than what we'll use today, but it still works. There were a lot of things Mendel didn't know about. He didn't know about chromosomes. He didn't know about DNA. He didn't know about meiosis. But the ideas that he came up with really fit with what our modern understanding seems to be. So first of all, alleles are the alternative versions of the genes that account for the different characteristics that are inherited. Okay, that's pretty easy to do. So you can have, for instance, in the case of the peas here, you have peas that are true breeding for yellow peas and peas that are true breeding for green peas, and those represent the different alleles, the different versions of the gene. For each characteristic, an organism inherits 2 alleles, one from each parent. The alleles can be the same or they can be different. Now again, he didn't call them alleles, he called them heritable characteristics or heritable characters, okay? But an individual that has identical alleles is called homozygous. The homo part means the same, and the zygous refers to the zygote that results from the combination of the sperm and egg cell. A heterozygous, hetero means different, genotype has 2 different alleles. So the 2 peas that are illustrated here in this picture are both homozygous for their trait. If we had one that had a capital Y and a lowercase y, we would call that a heterozygous one. All right, number 3, if the alleles are different, then one of them is going to determine the appearance. That's called the dominant allele. The other one doesn't really have much noticeable effect on the appearance and is called the recessive allele. What we call the physical appearance or the expression of the trait is called the phenotype. If you think phenotype, physical, that kind of helps you remember that that's referring to the expression of the trait. Sometimes it is something that you can see, as in the height of the plants that are shown here in the picture. Other times it's not. Okay, the genetic makeup of the trait we call the genotype. Okay, and that's whether it's homozygous or heterozygous. We can sometimes have the same phenotype determined by more than one genotype, as we see here in the case of these tall plants versus the one short one. We can have 2 different genotypes, the homozygous dominant and the heterozygous, both produce the same phenotype, even though they're different genotypes. Number 4, a sperm or an egg is going to only carry 1 allele for each inherited character because the allele pairs separate or segregate from each other during the production of the gametes. This is called the law of segregation. And this goes back with what we learned about meiosis in the fact that the chromosomes separate independently of each other as well. They're separate from each other as well. So this also explained the 3-to-1 ratio of the F2 generation, because the hybrids all have the F1 hybrids all have the heterozygous genotype, And so we can use a Punnett square to show the different combinations that could occur when you combine those gametes. So here we have our parent generation. We have our true breeding purple flowered plant and our true breeding white flowered plant. All of the gametes that can be produced by one are going to be identical. And so when those combine together, then all of the offspring are going to have one of each type of gamete. They're going to have a capital P and a lowercase p. When the F1 generation cross with each other, half of their gametes are going to be capital P's and half of their gametes are going to be lowercase p's. And so you can put those in a Punnett square that we use nowadays to show this, okay, to show what happens with the F2 generation. And so we have the letters that we place on the outside of the Punnett square here are representing the possible gametes that the parent could produce. And then the inside of each box within the Punnett square indicates the possible genotypes of the offspring. And so if this particular zygote received both dominant alleles, then that would be a homozygous dominant. This one receives a lowercase or a recessive allele from one parent and a dominant allele from the other parent. And that gives us heterozygous. Same thing here. And then if it receives both recessive alleles, then it would be the homozygous recessive. So our phenotype ratio gives us a 3-to-1 ratio of purple to white. That means 3/4 of their possible offspring would be more likely. It was likely that 3/4 of their offspring would have purple flowers and 1/4 would have white flowers. The genotype ratio shows a 1-to-2-to-1 ratio. 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. Now I'm going to give you a lot of problems to work. Okay, so we're going to work through some problems to see how that goes. Right? So here's an example of a problem. And this little GIF down here in the lower corner from the Amoeba Sisters kind of gives you an idea of how to go about doing this. And so you can watch that as it goes as well. So in humans, the ability to roll one's tongue is thought to be controlled by a single dominant gene. Two parents who are both tongue rollers each have one parent who is unable to roll their tongue. What proportion of their children can be expected to be tongue rollers? So the first thing we want to look at is the genotypes of the parents and the possible gametes they can produce. Since each one of these parents can roll their tongue, we know they have one dominant allele, but they each have a parent who is unable to roll their tongue. So that means that the two parents are going to be heterozygous. Okay? So what gametes can they produce? They can produce either a capital T, a dominant allele, or a lowercase t for the recessive allele. All right, we're going to put those on our Punnett square. Remember that we put the possible gametes on the outside of the square for both parents, for the father and the mother. It doesn't really matter which one you put where. I just said father and mother that way. Okay, it doesn't really matter. And then when we fill in the Punnett square, we're going to put the two letters that are on the outside of that box. So here we have a big T and a big T, so that gives us a homozygous dominant. Here we have a little t and a big T. There we have a heterozygous. We always put the capital letter first, the dominant allele first and the recessive allele second. Here we have another heterozygous. And then this final box here has our two recessive alleles for homozygous recessive. Now, one thing I'm going to ask you to do, and I'm not going to ask you in every single question that you have, I'm going to ask you a lot of times to show me the genotype ratio and the phenotype ratio of the offspring. Here's something really important that I want to get across to you while you're working on this, okay? Just because it shows up 1 to 2 to 1 or 3 to 1 in terms of the genotype or the phenotype ratio. That doesn't mean if those two parents have 4 children, they're going to have those particular results. What it means is every time they have a child, every single time they have a child, every fertilization event, they have 1 chance in 4 of producing a homozygous dominant, 2 chances out of 4 of producing a heterozygous, 1 chance out of 4 of producing a homozygous recessive. Okay? Just so you know, every Punnett square, every new fertilization event is a brand new Punnett square. Every time you start from scratch, every single time. All right, so the genotype here, the genotype ratio we have is 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Our phenotype ratio, remember this is the expression of the gene, is going to be 3 tongue rollers to 1 non-roller. Now, once again, if they have 4 children, they might have 4 tongue rollers. They might even have 4 non-tongue rollers. They might have half and half. But each time they have a child, there's 3 chances out of 4 that child will be a tongue roller, and 1 chance out of 4 that they won't be. All right, I'm going to give you several problem sets to work. The first one is called Introductory Monohybrid Crosses. That one's for practice, just to get you back in the swing of thinking about how to do this. The one that we'll really take a grade on is the one that's called More Monohybrid Crosses. I will provide for you Punnett squares and places to put your information. Some of it you may have to write down on paper and then transfer your answers to the worksheet to submit on Schoology. This is the end of the notes for part 1.